Okay.
Can you see?
Okay. Excuse me?
Can you see it?
Yeah.
Okay, cool. So it's you say it like shikimate, right? Am I saying that right?
So, Lindsay, this is like a word that I've been familiar with for a long time, but I've never heard anyone say it. So you know what I mean? So I call it shikimate,
Shikimate.
but
Okay.
yeah, but I'm not certain that's how like other people say it.
Okay, that's cool. All right, so this is the shikimate pathway. Weird. Um, so the shikimate pathway is a 7-step metabolic pathway um, used by archaea, fungi, algae, some protozoans, and plants. It leads to the biosynthesis of folates and aromatic acids. Aromatic amino acids like phenylalanine, tyrosine, and tryptophan. It's essential for the life of these organisms, but it's not essential for like humans, which makes it a great drug target because you can target their cells and like cause like cell death in those cells without harming the cells of the host. Okay, so this is the overview of the pathway right here, and I'm going to take you through step by step and just kind of like categorize each reaction. And then as you can see right here, this is charismate, which is the final product of the shikimate pathway. You can see over here, this is how charismate is used along with PEP and E4P to make phenylalanine, tyrosine, tryptophan, and other important amino acids essential to life. So the first step is an aldol condensation reaction in which phosphophenylpyruvate and D-ethyrose 4-phosphate we undergo a aldol reaction catalyzed by DAP synthesis to make a DAP molecule. This is probably one of the most important steps because at this step, many other like pathways are led off of this step. So yeah. And then this is the second step where it is, sorry, one second, which is the generation of the first carboxylic compound in which DHP is made into DHQ. And so it's a cyclic oxidation and reduction of NAD plus and NADH and the dephosphorylation of DHP, and it's catalyzed by DHQ synthase. So it starts with a C5 oxidation, which is bound by the enzyme, and then NAD plus binds, which triggers a cascade of spontaneous events. And then there's a C6 enolization, and then there's a beta elimination of the phosphate group, and then a reduction at the C5 regeneration NAD plus, and then the Pyranose ring opening, and then an aldol intermolecular reaction. And then step 3 is a dehydration reaction, and you can kind of see that the water comes from, you can see in this compound right here, how there's an alcohol, so it combines with the hydrogen, and then it's released as water, and then it makes DHS. It also involves an elimination reaction and enamine formation, but they didn't say much about where that happens. Step 4 is the NADPH dependent reduction. And so I don't know what your guys' bio background is, but NADPH is a electron carrier from the electron transport chain, and it basically carries energy and is able to donate electrons and can act as a nucleophile. So it's basically just a reducing agent, and it adds a hydrogen to this oxygen right here in this pathway.
It is exactly like sodium borohydride.
Okay. Yeah. Mm-hmm.
Well, then, in this context, it is. Yeah.
Okay. And then step 5 is phosphorylation and rearrangement, and this is starting on the right side and going to the left. And ATP is used as energy and ends up to dephosphorylate this molecule. This molecule, SHK. And then the 6th reaction is the addition and elimination reactions. And this is EPSP and S3P. I will be talking more about this specific step later on too, so just keep that in mind. And so you can see that the PEP is added to the S3P right here, and then it forms a tetrahedral intermediate. And then the elimination is when the phosphate group is removed from the PEP. And PEP, what's interesting about this step is PEP is also involved in the first step of the reaction. So this is the aldol condensation reaction where PEP and E4P react to form DAP. And it's just interesting because that molecule binds with the enzyme. So it's like kind of like you can create multiple inhibitors that will inhibit the pathway in multiple areas. And then the last step is a dephosphorylation, in which EPSP is dephosphorylated to form charismate, and then that charismate molecule is then used to make like tryptophan and all of those other compounds. So the connection with tuberculosis is a mycobacterial infection. There are multiple types of infections. It can be latent or active infections, and an active infection means you're contagious, you're showing symptoms, and latent basically means that the bacteria is inactive in your body. About 2 billion people have latent infections, and because people have latent infections, they end up being exposed to antibiotics, and because of that, it leads to antibiotic-resistant strains. And so they estimated that 400,000 people had the multi-drug resistant TB, which means that they are resistant to the first line of drugs, which is just oral antibiotics. And then 8.5% of those 400,000 people have extensively drug-resistant strains, meaning they are resistant to the injectable forms of antibiotics, which are stronger and have greater effects. So basically it doesn't work on them. And because of this, it makes Shikimate pathway a good solution to this problem, because they need new drugs for TB because there are antibiotic-resistant strains developing. And the Shikimate pathway is vital for TB to survive, but it's not for humans and other animals that may be infected with TB. So we can kill the TB without killing any of our cells. And I'm just going to do an example of an inhibitor in the Shikimate pathway, which is the glyphosate. I don't think I'm saying that right. Glyphosate?
Glyphosate.
Glyphosate, okay.
Yeah.
Which is the glyphosate herbicide, which is just an inhibitor example.
It's Roundup.
Yeah, Roundup. It's a weed killer.
Yeah.
Okay, so this is the enzyme, this is this sixth step that I was talking about that you guys kind of need to remember. This is right here where S3P is catalyzed into EPSP. And so the enzyme EPSP synthase is this enzyme right here. And here is S3P, which is this molecule. And then here's PEP, which the enzyme catalyzes that addition of the PEP to the S3P. And so what happens is glyphosate binds with S3P instead of PEP, and then basically inhibits that reaction. You can kind of see in this picture that there are lots of structurally similar parts of each molecule. They both have this tert butyl group, and yeah.
That's a phosphate group.
Oh, that's a phosphate group. Just kidding.
Yeah. Yeah, that's good.
Okay, and then I just kind of made this because I thought it was fun, but I don't know if it's going to work because I can't seem to figure out how this, okay. So glyphosate comes in, and then comes into this step right here, and then it prevents that reaction, and then it prevents charismate synthesis, so then charismate can't be used to make all of these, and then so that doesn't happen, and then they die. So
that was great, Lindsey.
Thanks. And then here's just a up close picture of this is S3P, this is F3P, this is the original binding PEP, and this is glyphosate, and you can kind of see how they structurally fit into each other. Yeah, that's it.
Very nice.
Thanks.
So does anybody have any questions for Lindsey?
This is like a dumb question, but what is latent?
There are no dumb questions, Emily. Go ahead.
I don't know exactly what the word means, but I know like a latent infection is when it's like inactive. So it basically means that you have the bacteria, but you aren't currently sick.
Okay.
Yeah, how I understand it is that tuberculosis can enter this state where it's like not growing, but still viable, which is what makes it so hard to treat. Because most antibiotics require the bacteria to be growing in order to work. So so these are basically insensitive to most antibiotics, and the people that have tuberculosis have to be on the drugs for like months and months and months. So yeah.
Thank you.
But this is, I mean, Emily, you probably recognize some of these molecules, so Lindsey mentioned that PEP, which is phosphoenol pyruvate, that many of the carbons that are in the final molecule, which is chlorismate, that's the molecule again that gets turned into all the amino acids and so on. So that molecule is like super abundant because it's a member of the glycolysis pathway. So like, it's just like a super common metabolic intermediate. And then the other starting material below it, that erythrose 4-phosphate, that's probably derived from glucose. It looks like that's just a small 4-carbon carbohydrate. Or it could be from like the pentose pathway. But anyway, these kinds of things are like sort of very accessible type molecules inside everything, really. The one thing that Lindsey probably is, or it's new to her and maybe many of you, is like what these reactions kind of look like. You know what I mean? Because there's like all of these molecules are basically functional group wise, they're carboxylic acids, alcohols, and phosphate groups. So it'll take a little bit of getting used to, I think, to see that. But the tricks are, I actually, like Lindsey mentioned, is there are some reductions which you can see because it uses the NADH or NAD as the cofactors. And interestingly, I thought there was 2 aldol reactions sort of back to back in the very beginning to make the first sort of cyclic version of the molecule. That's kind of an interesting thing too. It's probably a little bit hard to see, but because just these kinds of molecules are just like crowded with functional groups, and sometimes it's a little hard to see that. But anyway, Lindsey, sort of for you, what I was interested in is sort of you'll get some exposure to metabolism, but also the one enzyme in the pathway that I'm kind of interested in is shikimate kinase, which is, I guess, enzyme 4, maybe. Yeah, it's step 4. Shikimate phosphate would be the product. There we go. Step 5, I'm sorry. So shikimate kinase, which is the enzyme that puts that phosphate group on there. Mostly because I know, and you will soon know, how you can get that enzyme to work and measure it in a outside of the cell using a purified protein. so we can make that enzyme and feed it shikimate, which you can buy, and then we can measure the production of shikimate phosphate. And there are a very small number of known inhibitors of shikimate kinase. So what you'll do sort of by the end of our time together would be to propose some new structures and maybe even synthetic pathways to make them as shikimate kinase inhibitors. She's so tired. Anyway, okay. Does anybody else have any questions for Lindsey? Thank you, Lindsey. Anyway, okay. I'm feeling an enthusiasm gap this morning. All right. Mostly because you're all muted and I can't hear anything except for myself.
I can't figure out how to stop sharing this.
Move your cursor up to the top and a thing should drop down.
The top of what?
Just tap your
At
screen.
the top of the screen. Yeah.
Oh,
I can.
I got
Did
it.
you see it? Okay.
I got it.
Should I like start